Assalamualaikum warahmatullahi taala wabarakatuh. So for today presentation, I will present about my proposal. So my name is Muhammad Nabil bin Mazlan, metric number S67802. So for my title research, I will make about a thin layer chromatographic and high performance thin layer chromatographic fingerprinting analysis of Pereskia-Bleo leaves. So we have four parts for this proposal, which is introduction, literature review, methodology, and expected result. So for introduction, we will have six parts. So for the first part is a research background of Pereskia-Bleo. So it will tell a characteristic about a Pereskia-Bleo. So just like recent study have documented its biological activity, so it will supporting its traditional use. So communities in Malaysia commonly use Pereskia bleu leaves for its medicinal properties. So this the Spanish shrub with orange red flowers is claimed to be effective against diabetes, hypertension, and even cancer. So additionally, it has been used to treat inflammation, gastric pain, ulcers, and for overall body revitalization. And furthermore, um, Prescription leaves are used in decoctions and pastes for various ailments. Next is a tropical organism. So basically, Parascablio is a unique member of the cactus family, which is flourishes in tropical regions like Southeast Asia. So beyond its use as a vegetable, Parascablio also hold a significant place in traditional medicine. So consumed as a tea or decoction, it's believed just like to address various ailments, including a cancer, diabetes, and inflammation. So research supports this traditional use, revealing anti-diabetic and antioxidant properties in the leaves. And the leaves are drought tolerance adaptations that allow it to survive in warm, humid climates. The traditional medicinal uses of Price-Cablayo highlight the potential benefits of tropical plants for human health. So this is just a summary for tropical organism. So basically, for problem statements, we will tell about a particularly interesting plant that deserves a further study. So by figuring out these chemicals are in it and how they affect our body, so we can learn more about its potential health benefits. So this research will add to what we know about a Pereschia species in general and create a profile of the valuable chemicals it contains. So this information could be used to develop a new medicines in the future. Traditional medicines will suggest that this Pereschia blue may have many health benefits, but scientists needs to confirm this claim. So this research will use a technique called thin layer chromatography, which is TLC, and high performance thin layer chromatography, which is HPTLC, to identify this chemical in Pereschia blue. So for the significance of study, this is just summary of my proposal. So it will explore a promising but underexplored species and also establish a reliable method for analysis. And next, we will analyze the chemical profile of Periskia bleu to identify an antioxidant compounds. And if rich in antioxidants, so this Periskia bleu could prevent a chronic diseases. So validate traditional medicinal uses of Periscabio through a scientific research. And Periscabio has a potential as a natural source of antioxidants and medicine. So for the scope of study that we will focus on this research, so we will use a TLC and HPTLC to establish a unique chemical profile, which is a fingerprint of Periscablio leaves. So we will identify a major constituents like alkaloids, flavonoids, terpenes, and other secondary metabolites that exist in these leaves. Also, we will provide a standard reference for Periscablio samples for quality control, and other things, we will identify secondary metabolites and bioactive compounds that present in the extract. So for the main objectives for this research, we will have three of the objectives. So the first one is to extract a Periscablio leaves using a methanol. The second one is to profile the secondary metabolites of Periscablio using thin layer chromatography. And the third one is the same method. It is the same objective, but we will use an HPTLC, which is high performance thin layer chromatography. Okay, the next one is chapter 2, which is literature review. So, in this context, I will tell about what we will analyze and assisting research that related to my topic. So, For the first one is taxonomy of Perescablio. So it's basically just tell about arrangement of the family for Perescablio. So it has a family which is Cactaceae, which is cactus family, and genus which is Pereschia. Pereschia is a big one, not a Perescablio, because Perescablio is a specific one uh, which is a species that I make this research for. So the second one, which is botanical description. So Perescablio is a unique cactus species within the Cactaceae family. So unlike most cacti, it has leaves and is native to Central and South America. So it can grow up to 8 meters tall and thrives in tropical and subtropical regions. And young Perescablio has spines that disappear as it matures. So the leaves are thin, long, and glossy. It produces orange red flowers and yellow fruits with black seeds. And isolated compound. So isolated compound is not specifically we will um, have in this research, but we will use TLC and HPTLC fingerprinting, which is techniques that used to separate and identify different compounds in this mixture. And isolated compounds in future research, it will based on the results of the fingerprinting. So scientists will might choose to isolate this specific compound that appear in Perovskite blue. The next one is traditional usage and features. So this is just a summary of it. For food, it will consume raw in salads, which is used in Malaysia and China and used in teas. Medicine, used to treat a wide range of ailments, including cancer, diabetes, infections, and other disease. So other use, the spine used for natural fences and crushed leaves for water purification. And the next one is pharmacological features. So studies suggest Periscablio leaves have antioxidant properties that could help reduce oxidative stress linked to chronic disease. So the research shows promise for Periscablio as an antihypertensive agent, potentially lowering blood pressure by increasing urine output and promoting blood vessel relaxation. And the last one is a focusing one, which is TLC and HPTLC profiling. So TLC, an advantage of it, it will analyze a numerous samples simultaneously and faster analysis times and lower costs. For HPTLC, the advantage of it, it will have a wider range of profiling options for complex chemical makeup and and enhanced detection capabilities for a broader spectrum of compounds. And also, it has a faster and more efficient analysis, especially for multiple samples of this Perovskite blue. Okay, the next one is a chapter 3, which is a methodology. So, apart from all of my proposal, so for methodology, we will focusing on a thin layer chromatography analysis and high performance thin layer chromatography analysis, which is HPTLC. So for others, it is not important and not just like specific. We will be focused. So for TLC analysis, we will do a sample preparation, which is 5 milligram of paracetamol. We will extract is dissolved in methanol, and a TLC plate, a pre-coated silica gel plate is used as the stationary phase, and a mobile phase, which is a mixture of hexane and ethyl acetate. So we will divide to 70 and 30 ratio. So this ratio might be adjusted based on initial result. So sample spotting, a small amount of the extract solution is applied as a band on the plate, and the visualization, the developed plate is visualized under UV light at different wavelengths that we have learned in application organic chemistry. So for high performance thin layer chromatography analysis, so it has a little bit of advantage, which is more precise sample application, faster and better separation of compounds, less solvent usage, and faster analysis time. And we will be used to create a detailed fingerprint of the chemical present in the extract. And this fingerprint will be unique to Perovskite blue. The other one, this is just not important at all. Just like phytochemical test, we will test six of the secondary metabolites, which is alkaloid, terpenoid, steroid, saponin, tannin, and flavonoid. And FTIR analysis, which is infrared. So after using the HPTLC to identify potential compounds, so this FTIR analysis will be used to understand their structure in more detail. Last but not least is chapter 4, which is an expected result that I get for my research proposal. So basically, for expected result, it is based on a fingerprinting analysis. So we will get a TLC expected result and HPTLC expected result. So as you can see on the left side is the SPTLC result. So, and the right one is the SPTLC expected result of Pereskia-Bleu. So this is just an expected result, not a real one, because I did not yet do the experiments. Maybe on the future I will do this based on my research of Pereskia-Bleu. So this is the expected result that maybe I will get from my result from my research to do a thesis. So that's all for the summary of my proposal research of Pereskia-Bleu. So thank you.